All right, take it away, Bob. Want to introduce?
Okay, well, first, thank you everybody for attending this, our first outside speaker in our cognitive neuroscience series. When Professor Miller's finished, then we'll take questions, and I'm really, as usual, hoping we can get a lot of good questions from the grad students and the postdocs with the faculty, either mute or last. And with that, I'm going to make a really quick introduction. I've known Earl for a long time, and the word is thrown around casually a lot that people do groundbreaking research. But in his case, I think it really applies. I think he's done some of the most critical systems neuroscience work that I've seen, and it's been highly influential in understanding brain mechanisms of executive control. I'm not going to go into them all. I'll just say, for instance, the idea of mixed selectivity, pluripotent cells in the frontal lobe comes really from Earl. The idea that you can use oscillations to slot single units in terms of behavior, different elements in a working memory store, again, from Earl's lab, and many more things that he may or may not talk about. So I'm going to stop there, and I just want to thank him for taking the time to present to us and turn it over to Earl.
Thank you. So let me share my screen. I'll get started. Host disabled screen sharing, so we have to...
Let's see what we can do about that.
There we go, I got it.
Okay. Thanks, Alexis.
Okay, I assume you can all see my screen.
Joni Wallace just appeared.
How's your car, Joni?
It's good. It turns out that 4 exhausts gets you back across the Bay Bridge quicker.
Ah, well, mazel tov. All right. Well, thank you, everybody, and thanks for giving me a chance to talk to you. Thanks for Bob for the invite. My name is Earl Miller. And in case you'll know what I do, I study the neural basis of high-level cognitive functions. We do so in non-human primates, monkeys. Our main methods are multiple electrode recording in monkeys performing cognitive demanding tasks. This includes both arrays of electrodes in one brain area and arrays of electrodes in multiple brain areas, all to look at the micro and macro circuitry underlying the network dynamics of cognition. And so you hear about me talking about 2 different types of measurements: spiking activity from individual neurons and local field potentials, which are the average activity of local networks of neurons. For those of you familiar with EEG, local field potentials are like EEG, although in EEG, it's like you're listening to them in the next room. With LFPs, you're actually inside the room with your microphones. Otherwise, it's just different, same sort of signal. So today's topic is going to be working memory writ large. And you'll see what I mean in a moment. Working memory, the most common definition, is the temporary holding of information online to make it available for further processing. This has been studied by groundbreaking researchers like Alan Badley and Joaquin Fuster and Pat Goman-Rakesh. But the most important thing about working memory is not just that it holds information online temporarily. The most important thing is that working memory is not just short-term memory. The most important thing about it is volition. Working memory volition is what makes working memory special. And there's a point that Pat Goman-Rakesh pointed out in her work on working memories, that we have the capacity for top-down executive control over our thoughts. We can control what we think about, how long we think about it. We can control when to discard something from working memory. We can control when to make a decision, when to make our actions. Volition is a main mechanism, but it allows working memory to wrest control from the environment and turn it into our own internal drives. And top-down control is a major function of the prefrontal cortex. So in this talk, I'll be giving you a brief survey of some older results. And then we're going to go into a more in-depth look at some new data, including some yet unpublished data, and a model of working memory and beyond. So let's dive right in. About 20 so years ago, we started our first experiments trying to understand how the prefrontal cortex acquires the kind of top-down information you need for volitional control over behavior, you need for goal-directed behavior. And one of the first experiments we did was a study where we examined the role of prefrontal cortex in learning shape categories. Now for this experiment, we used 3 prototype computer-generated cats, shown right here, cat 1, cat 2, and cat 3, and 3 prototype dogs. And we used a morphing algorithm developed in Tommy Poggio's lab to morph or blend all these different shapes so there was a continuous smooth shape space, a smooth change from one morph to another. And you can see that down here on the bottom. This is an example of just a very small, we generate thousands of unique images. This is a small subset of just some of the images, but it illustrates the point that if you look across this morph line right here, this cat is gradually becoming this dog, and this dog is gradually becoming this cat. And what we did is we taught our monkeys that there was a sharp boundary that didn't actually exist in the real world. And that boundary is shown here at the 50-50 shape space. So anything more than that was more than 50% cat was a cat, anything more than 50% dog was a dog. And the monkey had to just tell us cat or dog. His job wasn't to report what the individual cat and dogs look like, just tell us which category that stimulus belonged to. And this allows us to disentangle the physical appearance of a stimulus, the fact that this cat looks very different from that cat or very different from this cat, disentangle that from the thing that the animal needed to solve a task, which is category membership. And all these cats belong to the same group, and all these dogs belong to the same group. And that's what we found. Now, here's just an example of one single neuron. It shows examples of each tile represents a cat stimulus or a dog stimulus that we use for that particular day of recording. The different morph lines are shown each row. So here's the cat 1 to dog 1 morph line, or this morph line right here. And here's the cat 2 to dog 2 morph line, etc. The blue shows the category boundary, and the color indicates the normalized firing rate, how much the neurons are activated to that particular stimulus. And what you can see here is that this neuron, and many others like it, it activated for all the different cat stimuli and did not activate for all the different dog stimuli. So the neuron is giving us a binary decision. The stimulus is either a cat or a dog. Now, again, so what this neuron is doing is throwing away any information about the exact physical appearance of the stimuli in this display and keeping only the top-down information needed to solve a task. That is, which group the stimuli belong to, group 1, cat, or group 2, dog. Okay. So in that experiment, we found that neurons reflected category membership, not exact appearance. We taught the monkeys these smooth, continuous shape group of stimuli belong to 1 or 2 categories. That's what we found in the prefrontal cortex, that representation. And the details of what the things look like, gone. When Andreas Nieder was in the lab, Andreas Nieder taught monkeys to recognize the small numbers 1 through about 6 or 7. And what Andreas found was that, after this training, again, Andreas trained the animals so the animals could recognize these stimuli and these number of stimuli in this display, regardless of how those stimuli were presented. And what Andreas found was neurons that reflected the number of items in the display, again, irrespective of their exact physical appearance. And when Joni Walsh was in the laboratory, what she did was train monkeys to switch back and forth between 2 different rules: match or non-match, or choose the same versus choose different. The monkey saw 2 pictures in a row, and if the monkey was following the match rule, the monkey had to pick the picture that looked just like the one it saw before. The monkey was following the non-match rule, pick a picture that looks different from the one it saw before, the non-matching one. And what they found was that neurons that reflected the task rules, whichever rule the monkey was following is what the neurons were signaling, the instruction on whether to choose the matching or non-matching pictures. So in all these cases, we see the same thing. We see by the time we get to the prefrontal cortex, we see a boiling away of the irrelevant details, like what the stimuli actually looked like, what these displays, the exact physical appearance of these displays, or for that matter, which stimuli the monkey was applying these rules to. All these different details were boiled away, leaving behind the essence of the top-down demand for the task. Again, consistent with the idea that what the prefrontal cortex does is absorb and utilize this top-down information for top-down control. When we started doing these studies many years ago, we found something very curious. we found that these effects occurred in about 30 to 40% of randomly selected PFC neurons. We do and don't pre-screen neurons for task-related responsiveness. Our methods are we drop our electrode arrays down into the cortex or wherever, and we record whatever neurons we can isolate. So we're randomly selecting neurons, yet we found something like 30 or 40% of randomly selected neurons in the prefrontal cortex showed these task-related effects. Now, many of you are maybe too young to remember a time back in the 20th century where the paradigm we all labored under was that every neuron had one function and one function only. And we could figure out the brain by figuring out what the function of each neuron does. Now, this actually caused a bit of a problem for that paradigm. Why are like 40% of the neurons, if each neuron just has one function, why are 40% of these neurons showing these effects? One possibility is that the task is overrepresented as a result of training. We train our monkeys for months on these tasks, so maybe the task has become abnormally largely represented in the prefrontal cortex as a result of this training. I find that very unlikely. Our monkeys have months of experience on these tasks. Humans have years and years of experience doing the same thing over and over again. Just ask Bob. Another possibility, and this is suggested sarcastically to me, one of the first talks I presented this work, is that, what are you saying? The monkey can learn 2 or 3 things and their brains would fill up? Well, no, that's obviously not true. But what John Duncan and I originally suggested, and we've carried forward with more recently with Stefano Fusi, is that many higher cortical neurons are multifunctional. They don't have a single function. The functional unit of the brain is not the individual neuron, but the ensemble. And individual neurons can have multifunctionality. They show what Duncan and I call adaptive coding, or more recently what Stefano Fusi and I called mixed selectivity. And the idea of mixed selectivity is mixed selectivity adds computational horsepower, something that Stefano and Mattia Ragati demonstrated in their computational modeling in collaboration with our data. And what mixed selectivity does is it creates a high dimensional representational space where a lot of information can be commingling. This allows solving of more complex problems, as Stefano and Mattia showed, is that if you have a network that can, where every neuron does one function, it only could solve very simple problems like make linear decisions, but only when you add in mixed selectivity can neurons then solve higher order, like nonlinear type problems. This allows greater storage capacity. You can use the actual activity from real data, which we had, to compute what is the maximum capacity of network storage of a network of neurons. The way to think about this is that if you have 1 million neurons and all the neurons do the same thing, you might as well have one neuron. So what Stefano and Mattia showed was that if you have a network where every neuron has one function, that network quickly reaches saturation. It only can learn a few tasks, then it can't learn anything else. But he started adding in these mixed selectivity neurons, and at some point, he added in about 1/3 of the neurons in the network showed mixed selectivity. He added in that many, and the network becomes essentially capacity unlimited. It allows greater flexibility and faster learning because all this information you need for behavior is all intermingling in the same population of neurons in this high dimensional space. So you can quickly learn arbitrary rules like stop at red and go green. And we found that since these studies, we found that this isn't just a function of the prefrontal cortex and very high level cortex. We can see examples of mixed selectivity throughout cortex. Mixed selectivity seems to be the rule in the cortex, not the exceptions, not just a function of the high-end stuff when you get to the prefrontal cortex. We're seeing it there's an apex where you get more mixed selectivity in the frontal cortex, but you can see it at all levels of the cortical hierarchy. And finally, just a quick plug, if you want to know about how the brain creates nice and stable representations inside this high dimensional space where there's a lot of activity commingling, there's a recent paper from Leo Kotskakov in my lab who showed mathematically and using known neural mechanisms how you can achieve these kind of highly stable representations despite all this high dimensionality and all this neural activity bouncing around in these networks. So mixed selectivity means that neurons participate in multiple ensembles. In other words, that the functional unit of the brain is not a single neuron, it's ensembles of neurons. But this merely points to an issue. If mixed selectivity means that neurons participate in multiple ensembles, it means what you have in the brain is ensembles lying on top of lying on top of ensembles lying on top of ensembles. There's lots of ensembles that share common neural elements with other ensembles. That's illustrated here, where I have just two ensembles, think X and think Y, and these two ensembles share a lot of common elements, what I'm calling here mixed selectivity neurons. Now, another vestige of the 20th century is that I learned in graduate school that anatomy is destiny. If two neurons are wired together and there's a strong synaptic weight, if one neuron fires, the other neuron will fire. Well, if this is true, how do you select a single ensemble from this overlapping anatomy? If I want to just activate the blue ensemble here, the purple ensemble, then if anatomy was destiny, if I activate the purple ensemble, the activity would run to the pink ensemble. I'd have both ensembles activated simultaneously, and my brain would be a mush. So how does the brain select a single ensemble given this overlapping anatomy that mixed selectivity implies? Well, one possibility is that neural ensembles are formed by synchronized rhythms. Idea first proposed by Wolfsinger, carried forward by Pascal Fries, Andreas Engel, and others. And the idea here is that, unlike you may have heard the neurons that fire together, wire together, which is the old Hebbian, how you build networks in the brain, we're suggesting that neurons that hum together temporarily wire together. Now, I'm not saying that wiring or anatomy isn't important. Wiring and anatomy is important for getting neurons to talk to one another. But if you want to select one ensemble of neurons from this overlapping anatomy, yet there's something else something else besides just anatomical wiring has to be at play. And the idea here is that you have these two ensembles sharing the same anatomy, but if the purple ensembles, if the neurons correspond to the purple ensemble, if they synchronize their rhythms together, that's what defines the purple ensemble, whereas the pink ensemble can either oscillate in synchrony at a different frequency or in different phases, something so they're out of sync with the blue ensemble, but in sync with each other, and then you have that ensemble. So this allows one ensemble to be selected from overlapping anatomy. And this may endow cognitive flexibility. You can change your mind. You can change it from one mode of thought to another. You can learn new things quickly. Humans can learn rules instantaneously. Flexibility is one of the defining characteristics of high-level cognition. And this provides a format for cognitive flexibility. Ensembles can be formed. They can be changed just by these shifting patterns of resonance. And what's nice about resonance is things, when they go into resonance, they tend to stay in the resonance until you do something to pop them out of resonance, then they can be disrupted and move to a new pattern of resonance. So the way I want you to think about this is anatomy in the brain is not destiny. Anatomy is possibility. It's like the road and highway system. Spikes carrying information are the traffic, and the patterns of oscillatory resonance are what directs the traffic. So with this in mind, when Tim Bushman was in the lab, he decided to study this on the prefrontal cortex of two monkeys that were trained to switch between two different rules. One rule was pay attention to the color of a stimulus. The other rule was pay attention to the orientation. So the monkey viewed a stimulus. It could be a vertical bar or a horizontal bar. It could be red, it could be blue. And at different trials, the monkey had to report either the color and ignore the orientation. And we set up the task in such a way that the monkey had to pay attention to just one dimension. Or the monkey had to pay attention to the orientation and ignore the color. And what Tim found is summarized here. Here, a representation of the lateral prefrontal cortex. The arcuate sulcus is here. The principal sulcus is here. This is posterior. That's anterior. The monkey's eye would be right here, looking forward. So lateral prefrontal cortex is about right here. The monkey brain, but you don't have a principal arcuate sulcus. The monkey does, but it's about right here in the monkey. Each of the circles is a different recording site. And the colored lines show which recording sites showed an increase in LFP synchrony, a significant increase in LFP synchrony for when the monkey was following the pay attention to color rule versus the pay attention to orientation rule. The color rule in pink and the orientation rule in purple. Now we looked for these patterns of synchrony. We looked for these LFT synchrony across all frequencies from 1 hertz up to 100 hertz. And we only found it in the beta range, about 12 to about 30 hertz. And what you see here is what is suggested by this idea of overlapping ensembles. One pattern of recording sites, a unique pattern of recording sites, go into increased beta synchrony when the monkey is following the pay attention to color rule. And you get a different but overlapping pattern of sites when the monkey is following the orientation rule. So this suggests that this beta synchrony is helping form the neural ensembles that form these top-down rules. Pay attention to color, pay attention to orientation. And Dimitris Pinosis in my lab, now on his own at University College London, has carried this work even further to look at the rules by which these ensembles form in real time. So if you want to hear more about this work, I suggest you follow up on Dimitri's fine papers. Now, once we had this result, we started doing new experiments naturally and looking over old data to see other examples of these kind of synchronous rhythmic-based neural ensembles. Evan Antolatis was in the laboratory. He trained monkeys to categorize locations on a computer screen as being up versus down. And what Evan found was that he found the evidence for these beta ensembles, both within the prefrontal cortex and between the prefrontal cortex and postparietal cortex for different spatial categories. One unique pattern of beta resonance, beta synchrony for one category up, and another pattern of synchrony for down. We went back and looked over our old cat and dog data, and we found the same exact thing. We found different patterns of beta ensembles when the monkey was making a judgment about cats, and a different pattern when the monkey was making categorical judgments about dogs. So one beta ensemble for cats, another beta ensemble for dogs. And finally, what Evan Anzalatis did, also along with Andreas Watts, is they actually trained monkeys to learn brand new categories over a 3-hour recording session, while we studied the same group of neurons as the animals were acquiring these categories. And what they found was that you can actually see these ensembles, see these beta ensembles form in the prefrontal cortex and between the prefrontal cortex and striatum as the animal is learning its categories. So the animal is, these dot categories were formed by these constellations, like you see here. Here's a prototype for category A versus category B. The monkey never saw the prototypes. The monkey saw distortions of the prototypes, as we call the category exemplars. And the monkey learned over about the course of like an hour or so by trial and error, which dot patterns were category A versus category B. And you actually saw these neural ensembles, these beta ensembles forming as the animal's learning the categories that exactly mirrored the animal's category acquisition. I think it's very compelling evidence that they're playing a direct role. These beta ensembles are playing a direct role in acquiring this category information. Okay, now with this in mind, let's turn back to the topic du jour. How do you hold thoughts in working memory? How do you hold thoughts in mind? Well, the classic model for the past 50 years or so has been persistent spiking of neurons. This is an example from the work of Pat Goldman-Rakesh, who was a real pioneer in these studies. And what it shows is the one neuron whose spike rate on the y-axis is averaged across multiple trials, and time is on the x-axis. It's a typical working memory task. You show the animal a cue. The monkey has to hold the cue in mind over a delay lasting 1 or more seconds. Then the monkey makes its response. And what's shown here is what you typically see in these studies. Their neurons get activated by the cue. When the cue goes away, activity drops a bit, but it maintains that activity above baseline over the course of the memory delay. Then when the monkey makes its response, activity drops back down to baseline. Very compelling. And it's a very compelling model, because what it suggests is you simply activate an ensemble, and that ensemble stays activated as long as you're holding it in a working memory. And I'm not here to tell you that's wrong. This is right. This model is essentially correct to a certain level of approximation. But more recently, advances in multiple electrode technology, which allowed us to study lots of neurons at the same time and analyze them in real time on single trials, has shown that in actuality, most neurons don't show this kind of persistent activity like you see here, like it's illustrated here on this figure. Most neurons in the population show very sparse and bursty spiking. They only fire a few times during the delay. And you tend to see this kind of evidence of persistent activity only when you average across lots of neurons and lots of trials. But on individual trials, you see very sparse and bursty spiking. And this has led to some updates to the classic model. One of which was from Paco and Rakesh herself, in one of her last papers, showed that spiking in the prefrontal cortex induces temporary increases in synaptic weight changes that Wong and Goldman and Rakesh speculated may carry the working memories between the bouts of spiking. There's also the activity silent model of Mark Stokes, which addresses this more explicitly. In the Mark Stokes model, you have neurons that fire periodically, then they stop firing, and they fire again. And in between the firing, the memories, the working memories are carried by these changes in synaptic weights that are induced by the spiking. And these changes in synaptic weights, they thought to rely on calcium dynamics, and they last about a little under a second. So you have to occasionally have spiking during the delay to refresh those synaptic weight changes. And that explains the sparse spiking during the delay. The point of all these updates is that spiking is not doing all the work. It's helped along by temporary impressions that spiking leaves in the networks. Now, for one reason, this makes a lot of sense, because spiking takes a lot of metabolic energy. And why would you have your brain evolve to be expending all this energy constantly as you're using working memory, which is pretty much, most of the time. What this shows is that the brain can save energy by using these temporary changes in synaptic weights to sort of carry the memories between spiking. But it also makes working memory more robust, because computational models of persistent activity using so-called attractor dynamics have shown that when you carry memories by persistent activity alone, they're really, really easy to disrupt. A distractor comes along. You add a stimulus to working memory, and that disrupts the original attractor state and disrupts the memory. These activity silent models, they address this problem by having the brain only be in this labile, easily disruptible attractor state only during these very brief periods while the neurons are spiking. The rest of the time, the memories are carried by synaptic weights, which are more robust interference. What I'm going to tell you about is a particular brand of activity silent models called the dynamic attractor network model, made by Michael Lundqvist. Michael was trying to explain how you can hold multiple items in working memory without having them interfere with one another, something that models of persistent activity have trouble doing. Now, one little point I want to make about this model is that Michael built this model from the bottom up using known biophysics, EPSPs, conduction velocities, IPSPs. He used known biophysics to try to build a model that could hold multiple items of working memory. And he had this model, it was all pure biophysical model. And then he called me up after he got his PhD and says, I have this new model that shows everything you've been doing for the past 30 years, or maybe it's 20 years at the time, 20 years, everything you've been doing for a long time is wrong about working memory. So can I come to your lab and use your data to prove you wrong? Of course, I was charmed by this, so I said, sure, come to the lab. And the point of this story is that Michael didn't build this model to fit existing data. The model was there first, and then we looked in data to see if the model was supported, and the model was very strongly supported. Virtually every aspect of the model we found support for in our data. So let me show you what it looks like. Here's an example of a single trial during a working memory task. We're now looking at LFPs. Frequency of the LFP is shown on the y-axis. Time is on the x-axis. In this particular working memory trial, the monkey has to hold two pictures, two stimuli in working memory. The monkey is shown the first picture here. There's a brief gap in time. The monkey is shown the second picture. The monkey has to hold two pictures in working memory over this delay here. Then the monkey makes its response. And in color is shown the LFP power at that particular frequency and time. Now, what do we see here? We see that there's these bursts of gamma that tend to happen during the encoding of the stimulus into working memory during stimulus presentation and occur occasionally during the working memory delay. What Michael found, which was consistent with the model, is that there's these bursts of gamma that happen during working memory encoding and storage. They're associated with the spiking that's carrying the working memories. All the spiking that's carrying the information about these two stimuli, they're all happening when these gamma bursts are occurring. So spikes carrying working memories are associated with the gamma bursting. What we also found, which is also consistent with the model, is that beta and gamma are anti-correlated. There were also bursts of beta. In Michael's model, the beta is kind of a default state. We'll see there's really no such thing as default state in the brain. We'll get to what I mean by default in a moment. In his model, though, beta is kind of this default state. And when there's a bottom-up input, like a sensory input, it activates the gamma bursting and spiking. And otherwise, when there's no gamma bursting and spiking, the network defaults back down to beta. And you can kind of see that here. Here's some gamma bursting here, here, and here. And down here in the lower frequencies, here's beta bursting. And the beta bursts tend to happen when the gamma burst is not happening. And that's better illustrated here, which shows now an average across trials of the rate of gamma bursting and beta bursting across time during this working memory task. Again, here's our two stimuli, stimulus 1, stimulus 2. Here's the working memory delay. Burst rate is on the y-axis. There's gamma versus beta, and they're mirror images of one another. Whenever gamma is going up, beta goes down. Whenever gamma goes down, beta goes up. So here, the working memory delay, there's a increase in gamma bursting during stimulus encoding. Then when the delay starts, gamma drops down, beta drops up. Then, as you often see in working memory tasks, there's this gradual increase in spiking and also gamma bursting during the as you reach the end of the delay. And as that gamma bursting and spiking is going up, the beta is dropping. Beta bursting is dropping. Now, I've said that the gamma bursting is associated with the spiking carrying the working memories, the contents of working memory, the two stimuli the monkey's holding in working memory. Gamma's anti-correlate with beta. What's beta doing? Well, that's the question. If gamma's carrying the working memories, what does beta do? Well, you may remember from the first part of my talk is that what beta is doing is carrying top-down information. When we train the monkey on all these tasks where we're looking at top-down demands, whether it's rules or categories are kind of like the same thing. We find that these top-down information is expressed not in gamma, not in other frequencies, but largely in beta. And we'll see in a few minutes also a bit in alpha. I'll get to that in a moment. So we tend to see that top-down information is carried in beta, whereas the contents of working memory is carried in gamma. Essentially, beta is carrying the rules of the task. The monkey knows the rules of the working memory task. That's being carried by beta, and gamma is carrying the content. And they're anti-correlated. So the idea here is that beta is like a top-down control signal. It has an inhibitory influence. It inhibits or gates the expression of gamma, the contents of working memory. So it provides a way, an infrastructure for top-down control of working memory. Top-down information carried by beta rhythms inhibits or gates the gamma carrying bottom-up information, the stimuli the animal's holding in working memory. It provides a way of turning on and off the faucet of working memory storage. Okay, now just to summarize one result, we took this further and we trained monkeys to perform a task in which is more complex in your average working memory task, where the monkey had to make a bunch of decision points. I'm going to spare the details of this paper because it's already been published. I'm just going to summarize the results. But if you're interested in the results, I encourage you to look up this paper. Basically, we found that this push-pull between beta and gamma reflects working memory control operations. So again, we have beta, the control, gamma is the content. Beta inhibits gamma. So we found that if you want to encode information into working memory, what happens is at the time of encoding, beta drops. That allows gamma to go up, and spikes start carrying the working memories. Then when you need to clear out working memory, you want to get rid of working memory because the memory is no longer relevant, the trial's over. What happens is beta goes up, that drives gamma down, and the spikes stop carrying the working memories. Then in one condition of the task, the monkey had to switch the contents of working memory from one stimulus to another stimulus, and you get this really cool rapid push-pull of beta and gamma. The first thing that happens is beta goes up, it drives gamma down, spikes stop carrying the working memory of the first stimulus. Now the monkey has to encode the second stimulus of working memory, and then beta drops real suddenly, and gamma goes up, and spikes start carrying information about the second stimulus. So you want to switch the contents of working memory. You first increase beta to clear the contents of working memory. It drives gamma down, working memory is cleared out. Then you want to gate the next stimulus in working memory. Beta drops and gamma goes up, and you've now switched working memory to the new stimulus. And we also showed that the balance between beta and gamma can predict the monkey's decisions and errors. We could tell why the monkey made a mistake. Did the monkey get the stimulus wrong? Did it get another decision point wrong? Did it mix up one stimulus from another? We could tell this by looking at the relative balance between beta and gamma on each trial. And we could better predict the monkey's decisions and errors with these beta and gamma dynamics than we could with spiking. Okay, so moving this work forward, we wanted to take a look at the local microcircuitry involved in these beta-gamma dynamics. So Andre Bastos and Roman Lunis, they started a series of experiments where they recorded from all layers of the cortex simultaneously using these so-called laminar electrodes, where there's multiple contacts along the shaft, so you can record neural activity from all layers of cortex simultaneously. This is relevant because we may remember from our neuroanatomy class that the superficial layers of cortex are the feed-forward layers of cortex that carry sensory information from the back of the brain to the front of the brain. Whereas the deep layers of cortex, they're the feedback layers of cortex that carry information, top-down information, from the front of the brain to the back of the brain. So the first thing that they noticed was that if you look at spiking activity related to working memory, it's mainly in the superficial layers of cortex. So here on the y-axis is the depth of cortex. We figured out where layer 4 was in the middle layer by using a current sink source analysis. Above it are the superficial layers, below are the deep layers of cortex. The color-coded is a multi-unit activity, the change from baseline multi-unit activity. And this is a working memory task that the monkey is instructed to hold a cue in working memory. The cue is shown here, and here's the working memory delay. And as you can see, the bulk of the spiking that's carrying the working memories are mainly in the superficial layers of cortex. This makes sense because the cue is a bottom-up input. It's a sensory stimulus. We ask the monkey to hold a working memory. So it makes sense that the cue would be held in the superficial layers of cortex because the superficial layers are the feed-forward layers of cortex that are carrying bottom-up sensory information from the front of the brain to the back of the brain. So if we're going to hold a stimulus in working memory, it makes sense it would be in the superficial layers of cortex. Then what they did is they looked at the relative power of gamma and beta in the superficial versus deep layers. So again, here's layer on the y-axis, 0 is the middle layer, or layer 4. Red is the relative beta power along the context. Blue is the relative gamma power. And you can see there, both gamma and beta power are present in both the superficial and deep layers of cortex, but not equally. There's more gamma in the superficial layers of cortex and more beta in the deep layers of cortex. Gamma power is higher in the superficial layers of cortex, and beta power is higher in the deep layers of cortex. This makes sense because remember, gamma is associated with bottom-up processing, bottom-up storage of sensory information. So it should be in the superficial or feed-forward layers of cortex. And I mentioned that beta is associated with top-down information. So it makes sense that beta should be in the deep layers of cortex. Those are the feedback layers of cortex that carry top-down information from the front of the brain to the back of the brain. So it's all making sense. Now this shows how beta and gamma are negatively correlated across layers. A minute ago, I showed you an example of how beta and gamma are anti-correlated. We found this anti-correlation, we being Andre Bastos and Roman Lunis, found that the beta and gamma are negatively correlated across the layers of cortex. So on the x-axis, we have which layer is providing beta, whether it's superficial layer of cortex or deep layer of cortex, or layer providing gamma, deep versus superficial on the y-axis. And the color is the power correlation. Red is positive, blue is negative. And the way to interpret this figure is that this anti-correlation we're seeing between beta and gamma is taking place across the superficial and deep layers of cortex. Whenever beta power is high in the deep layers of cortex, gamma is low in the superficial layers of cortex. Whenever gamma power is high in the superficial layers of cortex, beta power is low in the deep layers of cortex. So the same push-pull anti-correlation, but now taking place across the deep versus superficial layers. So now it seems that this top-down, bottom-up, feedback, feed-forward, beta, gamma interplay is taking place between cortical layers. The idea being top-down or executive information is carried by beta rhythms in the deep layers of cortex, and they have an inhibitory influence on the expression of gamma rhythms in the superficial layer of the cortex that traffic bottom-up information, that feed forward bottom information from the back of the brain to the front of the brain. So again, this way of turning on and off bottom-up encoding and working memory storage. So to sum up this model so far, we found that top-down information is carried by deep layer beta. Deep layer beta regulates superficial layer beta. There is some beta in the superficial layers. It's weaker than in deep layers, but it's there, and it's coupled to deep layer beta. Superficial layer beta, in turn, inhibits, gates, or controls gamma and the spiking that underlies working memory storage. So once we finished this round of investigations, we thought to ourselves, well, why should this just be true for working memory and frontal cortex? You see things like beta or alpha, which is the next frequency band down from beta. You see that all over cortex. You see gamma all over cortex too. Why should this only be true for working memory in the frontal cortex? So recently Michael Lundquist did a study where he looked at these dynamics all over cortex, in frontal, parietal, and visual cortex. And what Michael found was that we see the same sort of interplay, same push-pull arrangements between superficial layer gamma and deep layer beta all over cortex, with one little twist. All these frequencies, all the frequency bands, they actually increase in frequency somewhat as you move from the back of the brain to the front of the brain. So low gamma becomes higher gamma as you move forward in the cortex. And what's interesting is that alpha, which you may have heard, alpha is often invoked for top-down visual attention in the visual cortex. Alpha in visual cortex becomes beta as you move forward in frontal cortex. So they all exist along this continuum. So this suggests that similar mechanisms between these alpha, beta, top-down dynamics versus this push-pull relationship with gamma carrying bottom-up information, this may actually take place all over cortex. So recently we've turned this model to try to explain something known as predictive coding. Probably many of you know what predictive coding is, but I'll just briefly summarize it. Predictive coding is that the brain constantly makes mental models of the environment. Your brain is constantly anticipating what's the next sensory stimulus that's about to come along. Your brain needs to do this because it would be overwhelmed if it had to process all the sensory information that's flooding into your brain right now. It's just too much information for your brain to process. So the brain makes mental models of what's coming next in the next few hundred milliseconds. And this generates ongoing predictions of these forthcoming sensory inputs. This allows the brain to suppress processing of predicted inputs. If something comes along and your brain expected it, it's not very informative because you knew it was coming along, so your brain suppresses it. It doesn't get fully processed. Instead, unexpected inputs, known as prediction errors, they are fed forward to update the mental models, because if a stimulus comes along, if an input comes along that wasn't expected, by definition, it's informative because your brain wasn't able to anticipate it. So your brain favors processing of unexpected inputs. Now, most models, predictive coding is pretty widely accepted, but its neural implementation is not fully understood. And most models of predictive coding use specialized prediction error circuits, where at every level of cortical processing, there are certain circuits, specialized circuits, that compare inputs to predictions, either pass forward the prediction error or suppress the predicted input. But we think these beta-gamma dynamics provide a more parsimonious explanation of predictive coding. Namely, that prediction are carried by alpha beta from higher brain areas down to lower brain areas. These alpha beta, it carries the predictions of what the brain expects to happen next. And this inhibits gamma and spiking in the pathways that process those predicted sensory inputs. So essentially what the alpha beta is doing, it's inhibiting, pre-inhibiting the circuits in sensory cortex that would process a predicted input. And that means prediction errors aren't the result of some specialized circuits. Prediction errors are the results of gamma and spiking being expressed in pathways that have not been touched by the alpha beta inhibition. So just simply the feeding forward, the gamma and spiking being allowed to be expressed in pathways in sensory cortex unaffected by the top-down alpha beta predictions. But for this to work, these dynamics, alpha beta in particular, has to be able to target specific stimuli. I've sort of talked about this up to now as if alpha beta is just generally turning on and off the faucet of working memory. But for these mechanisms to be truly powerful, they have to be able to target particular stimuli, not just turn on and off storage in general. Well, they do. So what we did in this recent experiment, now under review, under revision actually, we did in collaboration with Nancy Capel's lab, is that we did laminar recordings in the prefrontal cortex, frontal Hemifields, LIP and 7A, and area V4. And the animal performed the working memory task under two different conditions. In one condition, and we used the working memory task, the monkey was always engaged with the stimulus. But in one set of conditions, the same sample object was used, as a sample for 50 trials in a row. So it was highly predictable. So like this car would be used for the same 50 trials in a row. Then the monkey switched to another block of trials where the sample object was relatively unpredictable, was chosen at random from a set of 4. So the monkey couldn't predict what the sample stimulus was. Now what we found is that alpha, beta, and gamma can indeed target specific stimulus representations in visual cortex. So for each recording site, actually this is, I'm just going to tell you about this one aspect of the data here. There's lots of stuff to chew on with this data because we had laminar recordings all over the cortex. But for this particular analysis, for each recording site in area V4 in visual cortex, we used the multi-unit spiking at that recording site to determine whether that site had a preferred object or a least preferred object. The most preferred object was the one that elicited the most spiking at that recording site. And the least preferred object was the object of the 4 we used that gave the least amount of spiking. Then we examined LFP power at that site. And what this showed is that the alpha, beta, and gamma effects were stronger when that site's preferred object was being used on that particular trial. So that's what's shown here. Here's now, these are LFP power in area V4. What we're looking here is that we're looking at the power on the y-axis. Now we've subtracted the predictive power during predictable samples from unpredictable. So going this direction, there's more power to when an unpredictable sample was used. Going this direction, there's more power. Below 0 would be more power when a predictable sample was used. So more power for unpredictable, less power for predictable. And here's the deep layers of V4. Here's the superficial layers. And what we see is that we see the highest increase in gamma in the superficial layers of cortex to objects that are not only unpredictable, but the preferred object for that recording site. So it's just that these dynamics are targeting specific stimulus representations. Now here's the same thing for the alpha beta predictions. Now we see the mirror opposite. We're now this direction going down. We see more power for predictable objects than unpredictable. And so, of course, for alpha beta shows the opposite of gamma, so we're seeing things happen in the opposite direction. And what we see is that there's the highest increase in alpha beta power in the deep layers of V4 versus superficial layers of V4. And we see the highest increase in beta. Actually, in this case, it's alpha beta, because now we're back in visual cortex. And now since there's this continuum from alpha to beta, we're now encroaching in on beta a bit, on alpha a bit. So you'll see me talk about alpha/beta now. In the deep layers of area V4, we see a bigger increase in alpha/beta power in the deep layers of the cortex when the object's being used is the preferred object for that particular recording site. So we see these dynamics can actually act on the representations of specific stimuli. Now, we also saw these effects played out in coupling between brain areas. Here's an example of LFP synchrony between the, in this case, frontal eye fields in area V4. Just for one example, we have in this paper, we have whole networks of areas shown, but for simplicity, I'm just showing you 2 areas. This shows the synchrony between V4 and frontal eye fields. And again, we have unpredictable minus predictable. So this is higher synchrony when the sample object was unpredictable. Here's higher synchrony when the sample object was predictable. And we see there's more gamma synchrony to unpredictable sample objects and more alpha beta synchrony when the objects were predictable, as expected from our model of alpha, beta, gamma being involved in predictive coding. Now, I'll mention one thing about theta here. You don't often see theta in monkey cortex. If you do, it tends to be very weak. When you do see theta, it tends to, it tracks along with gamma. So it's like 2 things. It's like theta and gamma do the same thing, and then between it is alpha, beta, which does the opposite. Okay, so we think this all provides evidence. Of course, this is not the first. We have more ongoing studies. But this provides evidence these alpha, beta, and gamma dynamics, these rhythms, not only support working memory, but also can support something your brain is doing constantly and automatically, and that is predictive coding. So finally, last topic I'll turn to is a study where we looked at the role of neural synchrony in actually unifying visual space. Now, we all know that visual cortex processing is split between the right and left hemispheres. So the right visual field goes to your left retina, your left visual field goes to your right retina. They cross at the optic chiasm so that the left side of your brain, left side of the visual field is processed by your right cerebral cortex, and the right side of the visual field is processed by your left cerebral cortex. And I probably don't need to say this to you, but I will because people often make this mistake. There's not a left versus right eye thing. It's a right versus left visual field thing. You get the same thing even if you cover one eye. So your one half of your brain processes the left side of vision. The other half of your brain processes the right side of vision. Now, we learned about this all in visual cortex when I was in graduate school, and it was widely assumed, almost like a tacit assumption, that now by the time you get to the cognitive parts of the brain, the prefrontal cortex, visual space on the right and left will be united. Somehow the prefrontal cortex somehow unifies space. Well, we've done those studies, and it turns out the split between the right and left hemifields remains up to and includes the prefrontal cortex and working memory. In fact, we did a number of studies showing that all of you probably know, you have a very limited capacity for working memory. You only can hold about 3 or 4 objects at best in visual working memory at any given moment. Well, we found in a series of studies that you don't have a capacity of, let's say, 4 is the average adult human. You don't actually have a capacity of 4. You have a capacity of 2 on the right and 2 on the left. And they're absolutely independent. So if you present a stimulus on the left side of the vision, it has no effect on neural processing on the right, no effect on the capacity on the right, and vice versa. As if monkeys and humans are all split brain patients. And in case you're wondering, we showed this in monkeys. Ed Vogel has followed up this work in humans and has found the same split in several thousand human subjects now too. So visual cognition is split between the right and left, but visual cognition seems seamless. So how does the brain unite the left and right sides of, I mean, you don't notice the split between right and left when you're playing Frisbee, for example. How does the brain unite the left and right visual fields? Well, we sought to begin to investigate this in a recent experiment where we did a little behavioral trick to see if we can get the monkeys to switch working memories between one side of the brain and the other, between the left cerebral hemisphere and the right cerebral hemisphere, and vice versa. And the way this experiment worked is that the monkeys start by fixating a target on one side of the computer screen, on the left or on the right. In this case, it's shown on the left. And then we load a stimulus in the working memory in just one visual hemifield. So in this case, the banana is being loaded into working memory in the right visual hemifield. Then half of the trials, we call the location swap trials, the monkey is instructed to make a saccade to the other side of the computer screen. So the monkey switches its gaze from here to here during the memory delay when there's no stimulus on the screen, but the monkey's holding the stimulus in working memory. This saccade, notice that this saccade would have moved the stimulus to the other hemifield, in other words, to the other cerebral hemisphere, if that stimulus was still on the screen. But it's not, the monkey's holding it in working memory. So the first question is, does this little behavioral trick, when the monkey's moving its eye while watching a blank screen, does it actually move the working memory from one cerebral hemisphere to another? And the answer, as you might have guessed, is yes, or I probably wouldn't be telling you about this experiment. But here's the predictions. So on top here, what we have is we're recording from both the right and left prefrontal cortex simultaneously. So a stimulus can either be contralateral to the recorded hemisphere or ipsilateral to the recorded hemisphere. And the working memory traces are stronger, way stronger for the contralateral stimulus than the ipsilateral. That's a split I was talking about. And the dark lines here shows what happens when a stimulus is presented on the contralateral side of visual space to the recorded hemisphere or the ipsilateral side, and they just stay there. There's no saccade in the middle of the delay, and the stimulus presumably just stays where it is. And what we see is what you expect to see. There's more activity for contralateral stimuli than ipsilateral stimuli. Now the lighter colored lines shows what happens when there's a saccade and the stimulus shifts from the contralateral field to the ipsilateral field, or the other way around, ipsilateral to contralateral. Now one possibility is that the saccade we had the animal make had no effect at all. Didn't mean anything to the animal. The memory just stays in the original hemisphere. In that case, we expect to see this. For contralateral stimuli, they'd be a stronger representation than ipsilateral. And it doesn't make a difference whether they make a saccade or not. The contralateral stimuli evoke stronger working memory effects, stronger working memory physiology than ipsilateral. The other possibility is that the memory does switch to the other cerebral hemisphere. In that case, we'd expect a switch. When the stimulus switches, when there's a saccade shown here by this gray line, when it switches the stimulus from one side to another, there should be a switch, whereas a stimulus that was contralateral before switches to the ipsilateral hemisphere, and therefore its representation gets weaker. Or in the case of ipsilateral to contralateral, it starts on the ipsilateral side, but then it switches to the contralateral side, and now the memory gets stronger because it's on the contralateral side. Well, that is exactly what we found. So here's an example. This is spike rate now, mean spike rate. Here's the delay before the animal has made the saccade in the middle of the delay that moves the stimulus. We see stronger spike rates, higher spike rates for contralateral and ipsilateral stimuli. Here's where the saccade is instructed. When the saccade happens, there's lots of neural activity because the eye's moving. Then the brain settles down, and look what happens. Here's the contralateral stimulus here. Now after the saccade is made, now the activity drives down to the same level that it would be if the stimulus was originally presented on the ipsilateral side. And the same thing for ipsilateral. Here's the stimulus starting on the ipsilateral side, very weak, almost no working memory representation. The saccade occurs, which switches the stimulus to the contralateral side, and boom, the activity now goes up to where it would be if the stimulus originally started on the contralateral side. So we do see the switching. The memory traces disappear from the sending hemisphere and reappear in the receiver hemisphere. Now, around the time of this memory transfer, there was a change in synchrony between the right and left cerebral hemispheres in the same bands I've been talking about all along. Namely, that around the time of the, here's time on the x-axis, here's frequency on the y-axis. Now we're looking at LFP synchrony, measure of synchrony between the right and left hemispheres by their LFPs. And around the time of the working memory transfer, there's a decrease in beta synchrony, that control signal, and an increase in gamma theta synchrony between the hemispheres. As if what's happening is if the brain is now pushing the memories from one cerebral hemisphere to another by using these alpha, beta, gamma theta dynamics. And sure enough, when we do a grain truth causality measure of LFP synchrony, we see that below 40 hertz, where all this magic is happening, the directionality of the synchrony is consistent with memory trace transfer. The directionality is higher in the direction from sending to receiving hemisphere than from receiver to sender hemisphere. So it seems that the one way the brain unites working memory is by moving the working memories back and forth in the cerebral hemispheres as needed, and it does so by the aid of these alpha, beta, and gamma dynamics. So to sum up, alpha beta is like a control signal that feeds top-down information through the cortex, through the deep layers of using the deep layers of cortex. It regulates the expression of gamma in superficial layers of cortex that traffic bottom-up sensory information and control, and also working memory storage. And what we suggest here is this interplay between deep layer, top-down alpha beta, and superficial layer feed-forward, bottom-up gamma. This is actually a modal cortical circuit that's at play all over cortex. And it plays a role in top-down control of lots of things: working memory and attention. Attention is on the other side of this, same side of the coin as working memory, just a different manifestation. And even predictive coding can play a role in predictive coding, and even the transfer of memories from one cerebral hemisphere to another when needed. So that's it. Thank you for your attention, and I'm happy to take any questions you might have. Should I unshare my screen, or how should we do this?
Yeah, maybe unshare your screen and we'll start getting some questions.
Okay.
Well, thank you very much.
Thank you.
Very much. Yeah, Christina, please.
Yeah, it's great talk. I just had a question about what you think like the role between this kind of like alpha, beta, and gamma is. I feel like some of the ways you're kind of talking about it almost sound like there could be a causal relationship between the two, but I wasn't sure if that was just kind of an easy way to explain it. Like if you actually think this role would be causal, or do you think that there's these two different systems?
Yeah, I think that's a great question. We think it is causal in the sense that it's a unidirectionally causal, whereas alpha beta inhibits gamma, but gamma doesn't have any effect on alpha beta. We've actually shown this physiologically in our data. We can see that when we look at synchrony measures between alpha beta and deep versus superficial layers. Alpha beta pushes around gamma, and gamma has no effect on alpha beta. So it is, we think it is causal, and we think it's unidirectional. We're now planning a series of studies where we're going to directly manipulate the alpha beta rhythms and see if we can push around gamma and behavior in a predictable fashion.
Cool. And what do you think the mechanism is for the beta to inhibit the gamma?
That's a good question. We're working on models of that with our collaborator, Nancy Capel. It may be a matter of changing EI balance. It's hard to say right now. It'd just be all speculation at this point, but it could be a matter of moving around EI balance. Just that what happens is that when you see these alpha beta going on, what you see the alpha beta is slow, and then the gamma tends to occur in bursts whenever the alpha beta bursting is low. And what's interesting about, I should mention something about theta now we're talking about it. In the Michael Lundquist model, the role of theta is to regulate the expression of gamma. So we don't see much theta especially in prefrontal cortex, but when we do see theta, the way we do see theta is that gamma bursting seems to be correlated with theta. So according to this model, what's going along is that you want to separate out these bursts of gamma in time, because each burst of gamma may underlie a different working memory. And theta helps regulate when gamma is going to burst, and therefore it keeps the working memories apart from one another in time.
Mm-hmm.
Okay, how about,
and then,
oh, I'm sorry. Go ahead, Christina, please.
Just the last kind of follow-up. When you're talking about the beta and alpha in the monkeys, do you think that is a similar kind of frequency band in humans, or do you think it's slightly slower or faster?
We think so. I mean, it's possible. Sometimes these rhythms are higher. Like, for instance, we're doing some studies on the effects of why general anesthesia makes you unconscious.
Mm-hmm.
And in humans, there's something called anterior frontalization, where you get this big increase in alpha in the frontal cortex when a drug like propofol first hits the nervous system, around the time of unconsciousness. We see the same thing in monkeys, only it's slightly higher in frequency. And that may be just a matter because the monkey brain is smaller than the human brain. Simple as that. These are all based on duty cycles and recurrent connections and so on. So it makes sense that the frequencies might skew slightly higher in a smaller brain. I will mention I'm sort of lumping alpha and betas together for good reason, because we see alpha gradually becoming betas and move forward in the brain. But some people, and this may also be true just under different functions, alpha and beta may have slightly different roles in different circumstances. And I just mention that because some of my colleagues like Nancy, who have very different ideas for alpha and beta, tend to get apoplectic when I lump them together. So that's just my disclaimer.
Thanks.
Can I ask a little bit related question? If alpha beta is inhibiting gamma, then do you have any guess on what might inhibit alpha beta activity?
What controls alpha beta? That is a great question. That's the million-dollar question of top-down control. How do you actually get top-down control? If I had the answer to that question, we could all go home because we have the brain figured out. That's the deepest question in neuroscience. Now, if you want me to wave my hands and talk about how it happens, well, the alpha beta, remember, it's this pattern influence from the front of the brain to the back of the brain. That's the way in which alpha beta controls patterns of expression of gamma. What controls that alpha beta? Good question. We know there's these loops between the prefrontal cortex and the basal ganglia. We see that we've seen the role of the prefrontal cortex and basal ganglia create these top-down representations through repeated iterations through these basal ganglia loops. And that's all gated by dopamine. So there's some hand-wavy thing about your brain learns to change representations in these loops through activating dopamine, which is your brain's pleasure, reward signal. And somehow that through a recurrent recursive process, you build up these representations and now exert control and direct you towards goals. Doesn't really answer the question, but that's the best I got at this point.
Yeah, thanks.
Thank you. Jacob, please.
Oh, thanks, Bob. Hi, Earl. I had a quick question for you, or maybe not so quick, you can see. But this is more about the first half of your talk when you were talking about the 30 to 40% number that you mentioned about kind of if you just randomly drop and record from a unit in lateral PFC, then about 30 to 40% and then will be selective for whatever the animal's trained to do.
Right.
And my question about that is, you also kind of mentioned offhand that you don't think that training would really have a big influence on this. But I have a question on that because there's been some studies, so like Christos Constantinides had a study last year where they kind of trained before and after monkeys did a spatial working memory task, and they showed that there at least was some increase in the percentage of cells that were selective. And so sure. I'm wondering, yeah.
No, go ahead, finish your sentence, please.
So I'm wondering kind of, if you were able to hypothetically record kind of right at the beginning of the animal's training, kind of what would you guess that the kind of number of, would you guess that it's a lower number of cells, and kind of if by how much that are selective? And then also that if you've done any experiments where you kind of try to then kind of once monkeys have learned this task, switch to some novel stimuli or something to kind of show that, it's not just the overtraining on a small number of stimuli, for example, that it's...
Let me correct something here.
Yeah.
When we do these experiments, we use new stimuli every day. So it's always novel stimuli. It's not...
Yeah.
We don't want the animal to just memorize a lookup table of stimuli. No, I mean, no doubt there's going to be an increase in the number of stimuli as a result of training, and the brain's got to learn. So you're incorporating new information in the brain. Of course, that's going to increase the number of neurons that do whatever you teach the monkey to do X, whatever you teach the monkey to do. What I'm saying is that, so of course, and when we did that categorization study where the monkeys learning those dot categories, you start out with 0 neurons that represent the categories, you end up with lots of neurons that do the categories. My point is that this notion that the huge proportion of neurons, 30, 40%, that large proportion is due to like overtraining is what I'm arguing against. I don't buy that at all. And even in that category experiment, overtraining, the monkey's learning 2 brand new categories every single day over a 3-hour session. That's not overtraining. And yet you reach that like 30% mark by the end of 3 hours.
Okay, that makes sense. And then if I could ask one follow-up question. So what's the limit on that number? So why isn't it like 80%, for example? Do you have any guess as to why that might be?
That's a computational modeling question. You computational modelers should answer that. I will say that in Stefano's, what Stefano did with his model in the Nature paper was that he took a network model that every unit did one thing, then gradually started increasing the portion of mixed selectivity neurons. And you get to some magic points where all of a sudden you get all the nice benefits of mixed selectivity. You get model can solve complex things, capacity unlimited. And this may be a coincidence, it's got to be addressed computationally, but that magic number he found was about 30 to 40%.
Thank you.
Okay, great. So we're going to go Albert, Willa, and JT, and then David. He's faculty. So sorry, David, but you're last. Albert, please.
Yeah, thank you. It was an amazing talk, and the whole thing about theta, the whole trade-off between alpha, beta, and gamma really got me thinking a lot. And I have two questions on that. The first one is, yeah, from a grand scheme of things, it seems like the alpha and beta, the slow waves represent, like you said, a global loop that seems to involve multiple brain areas. Mm-hmm. And then whereas gamma frequency band is more of a fast transient representation of the information. And this actually ties, and so far you have looked at very extensively in all those higher level or the lower cortical areas. And but I got a feeling that this kind of principle might also apply in areas like striatum, where you have reward prediction errors, which are fast transient information. And if you record it with alpha p, that might just be represented as a increase in gamma power. And then you have the tonic theta beta band. That's the first question. And the second question, or I can ask like after you finish.
Let me do the first one first. Yeah, predictive coding is more of cortical function. So yeah. we haven't done these experiments in the striatum. It'd be interesting to do it in the striatum, but we may find very different dynamics there, at least when it comes to like predictive errors and prediction.
Yeah.
Is that your question? We haven't done it.
Kind of.
I will say that there's a truth to one thing you said, is that alpha beta seems more of a global thing, whereas gamma is more of a local transient thing. And that's actually that's kind of true in our data. Gamma is more of you get feed forward synchrony in the gamma, but gamma is more of it seems to be less of a global phenomenon than a local phenomenon that's passed forward. And one evidence for this is we have a brand new paper we're working on right now, where we find that these theta, alpha, and beta, these oscillations, they don't just stand there and oscillate, they form spiraling waves in frontal cortex. They actually spiral around. And you can see them spiral around in a task-dependent way. You do not see that for gamma. So gamma seems to be a different animal from everything below it.
Okay, so let's, we got two more.
I think you have one more question.
Oh.
Yeah, can I add on the second question? Yeah, thank you. Yeah, so with that sort of brings up a bigger question, and I think Jacob might have already touched upon it a little bit. So the alpha and theta band, alpha band, beta band, and gamma band, they are sort of emergence phenomena once you have this multi-unit recordings with LFP, and they are essentially like the representation of like integrated levels of different neurons activity. And then we sort of get to this like weird loop of chicken and egg problem where we can say that alpha and beta suppress gamma, or we can say that gamma it's because of this local increase that disrupts the global activity pattern. It's not like, yeah, so
sure.
Yeah.
I see what you're saying. Yeah, it's important to keep in mind that underlying all these dynamics are all spiking, right? I mean,
right.
There's spiking underlying alpha, beta, there's spiking underlying theta, there's spiking underlying gamma. When I talk about the spiking in gamma, I'm specifically talking about the spiking carrying bottom-up information. The spiking carrying the rules is associated with alpha beta, not with gamma. Okay, but you're right. It's not really a chicken egg thing, because keep in mind that spiking and LFPs, they're the exact same phenomenon, just studied at different levels of integration, right? So you can't
Right.
separate one from the other. They're not two separate things. They're the same thing, just studied in different ways. When you study the level of P, you're getting more, because you're sampling more neurons, you're getting a better idea of what these network dynamics are like. Whereas when you study the level of single neuron, you're looking at information processing in individual neurons. You can't see the dynamics as well because you're focusing in on the individuals, but they're all manifestations of the same thing.
Yeah, but I mean, the
Albert,
beta.
Albert,
sorry.
Albert, I got a bunch more people. So if we have time, we'll come back. We'll cycle back, okay? Willa, Willa, please?
Yeah, thanks. Yeah, so that was a great talk. I had a question more about, so you mentioned the basal ganglia, but I was also wondering how you think the sort of the dynamics from the hippocampus or something might influence the predictive coding that's happening in the prefrontal cortex, and how those might affect, both the beta but also any gamma oscillations.
Yeah, good question. Don't know the answer yet, because we haven't studied it yet. I mean, we do know that we think that the frontal cortex and basal ganglia work together to sort of bootstrap top-down representations. And we've seen examples of this where what the prefrontal cortex is doing is taking the things that the basal ganglia learns and turning it into a top-down representation. We did a study a while back, Scott Brinken in my laboratory did a study where he looked at the animals learning new association-seeing stimuli, and he recorded from the hippocampus as well. And what the hippocampus seems to do is provide an error signal to the frontal cortex, at least in that particular learning paradigm. The hippocampus is saying, no, you got that wrong, no, you got that right, providing this, error signal to the frontal cortex. How that may play a role in predictive coding, I don't know yet, but I'm pretty sure it would, because hippocampus is all about keeping an ongoing record, at least temporarily and maybe permanently, of ongoing experience. So I would imagine that it would help feed into these predictive representations. That's something we need to address experimentally. I don't know anybody, all the models of predictive coding I know about are all cortical models. I haven't heard much talk about the hippocampus, but I think your instinct is right that it must also play a contributing role.
Yeah, I was thinking it would play a role in some sort of like context or rule learning, but that's cool. Thank you.
Sure, thank
Okay,
you.
so we have JT and then David and then Kevin. JT, please.
We also have Shamak with a question. He's.
Oh, yeah, I'm sorry. Yeah. Oh, there's your hand. Okay, good. Sorry.
Great. So I just had a question about, so you talked a lot about how working memory could maintain representations and then you can make a judgment like cat or dog. I was wondering, do you have a theory about like mental rotation, right? You have a representation that's being like manipulated in a continuous way. So how does this alpha gamma or beta gamma kind of facilitate that process in prefrontal cortex?
Great question. Don't have a detailed answer for you right now. I can say that we think the alpha beta can not just turning on and off working memory, but it's involved in the manipulation of items in working memory. I'm just saying that because it seems to provide this top-down signal. And the one manipulation we did have in one study, when the monkey has to switch the stimuli working memory, switch from stimulus 1, get rid of it, code in stimulus 2, and we see these alpha, beta, or in this case, beta gamma dynamics do that switching operation. How that would work for a gradual change in representations, I don't know yet. I imagine it could be just a gating of different representations along that rotational space that can be gated by the alpha beta, by the beta dynamics. I just don't know the details now. We are, Dimitris Pinosis, who was recently in my lab, I'm still collaborating with him. He's at UC London now. We're working on these kind of questions right now. How you can use these dynamics to not only gate information, but also manipulate it. So I don't have an answer for you yet, but I hope to have an answer for you in the future.
Great.
Great. Sarmak?
Yes. Hi. In the first part of your talk, how did you define an ensemble? You identified these various ensembles. What was like the criterion that you set to call something an ensemble versus not?
I didn't define an ensemble. I said it looks like, I said these beta dynamics look like an ensemble. And what they look like is they're not just, what I showed you is individual pairs of recording sites showing greater synchrony. But what I didn't show you from that paper was you can do a network analysis and show it's not just individual pairs of recording sites showing this increase in beta synchrony for one rule or the other. It's actually constant patterns of activity, all different shape for different rules, but they're actually whole patterns of activity. And you can do a network analysis to show things like how tight the network representation is for one rule versus the other. The harder the rule is, the more the network dynamics become compact. So there's a whole bunch of things that show, that indicate that these are actual active networks that are corresponding to one particular top-down representation versus the other. Now, how to define a ensemble in the actual data? This is exactly what Dimitri's is working on right now from these kind of data. Right now, I've sort of treated it as, here's pieces of an ensemble, and together they look like an ensemble to me, but this is the kind of, Dimitri's is working now in our data to try to figure out what are the mathematical rules, how can we use these patterns of synchrony to actually make it at least an operational definition of what an ensemble is.
Okay, so let's go to, I've got David, Kevin, and then I think Albert has one more question. David, please.
Hi, Earl.
David, how you doing?
Good to see you. Yeah.
See you.
So I have a question from the hippocampus came up already. So I have a question from the rat hippocampal world. We have this idea of high and low gamma, and where high gamma is associated with memory encoding, and low gamma with memory retrieval. Perfect, right? Because the encoding would be like bottom up and the retrieval would be, I guess, top down or inside out.
Yeah,
And
when you say high and low, what frequencies are you talking about?
yeah, so low gamma is like from 25 to 50 hertz and high gamma is like 70 to 100 and something. And so it overlapped with at least some of your, so is that just, you know, we say potato, you say potato, or is it a substantive difference, you think?
No, I think it might be, because there is, if you look, like, low gamma seems to be a little bit different from high gamma. Okay, high gamma is really, when you see things about 50 hertz, that's where the encoding really takes place. Like, but for example, you notice that last thing, the memory transfer thing, that gamma was pretty low gamma. It was like around 40 hertz. So I think that may very well map onto what you're finding in the rat hippocampus. I'd like to hear more about that, please.
Yeah, let's chat.
Okay, so let's go to Kevin, please.
Thanks, Earl, great talk. So I'm wondering, you know, in your early years in grad school, you spent a lot of time thinking about repetition suppression versus repetition enhancement. And I'm wondering how that factors in, or maybe it doesn't, or you haven't thought about how that factors in into the present models that you're pitching with working memory.
Wow, I hadn't thought about repetition suppression in like 30 years. Jeez. So the answer is I haven't thought about it. No, I mean, like, the repetition suppression seems to be, I mean, that may play more of a role in the predictive coding than anything else. Is that what's going on there? Do you have a stimulus?
Yeah.
And I think that's working memory, not so much. Working memory, you're holding something in mind, whether it's familiar or whether it's, but the repetition suppression and the gradual decrease in activity with familiarity, that seems to be more of an automatic memory thing, automatic recognition memory thing. So I fully expect that would be part and parcel, may be affected by these alpha beta dynamics, and may very well be one way in which the predictive coding mechanism takes place.
Cool. Thank you.
Thank you for reading my work.
By the way, before I forget, Rich Ivory had to leave for a Zoom meeting, and he says, great talk.
Oh, thanks,
so
Rich.
Back to Albert. Where are you, Albert? Did you have a follow-up question?
Yeah, thank you. Yeah, thanks. Yeah, and for the second question, and sorry, before I didn't really like say it that well, but it's not very important. Like after I think about it a lot, yeah, the second question wasn't really a question, but now I had another question that I always wanted to ask someone who is very well versed in brain oscillations and all those computations. So in quantum physics, people use the Schrodinger's wave equation for computation, and And with Heisenberg, he developed those matrix operations on top of those wave functions. And they have sort of this two different formulation of how things sort of work. Do you know, are you aware of anyone who might have adopted similar mathematical concept into the studies of brain functions, where they use these brain waves to represent, they use Schrodinger waves to represent brain waves, and they use matrix algebra to manipulate this like rotation, as Jonathan has mentioned?
Yeah, you lost me at quantum physics. I don't know. Maybe it goes back to Newton or Einstein, where, you know, you have waves and spikes and they're like, well, they're both waves and particles at the same time. I don't know. You know, I don't know. You know, I work with a couple of physicists. Dimitri's is a physicist, for example. We haven't thought about it in those terms. That's an interesting suggestion now. Hey, if you want to, you know, think about it and talk about it with me offline, let's do it.
There you go,
Thank you.
Albert. You got your own personal gateway to the frontal lobe. So is there anybody else who has any questions for Earl, who's been very kind with his time? Anybody? I don't see any hands going up. Okay, so Earl, the worst, let me just say, I've heard you talk many times, and each time I learn more, and you provide more insights into how behavior is enabled. So congratulations. Thank you for spending time with us. I'm only sorry we're not going to have a reception for you and be able to take you to one of the
Where's my wine?
place for dinner. So we owe you one. And
Okay.
when this COVID's over, we'll deliver the goods for you.
All right, deal.
And the other thing I just want to say to everybody, we're going to post under files on the course. Earl's lecture will be there. I know for some of you who haven't heard him before, there's a lot of material, but you can go through it. The papers are really very clear and elegantly written. So unless there's any other suggestions or comments from anybody? No? Earl,
Thank you. Thank you very much.
Thank you.
And folks, the paper is
Thanks, Earl.
Working Memory 2.0 is posted on B courses. If you want a copy, let me know. Thank you again, Earl, so much.
Thank you.
Thanks, everybody.
Loudly.
Bye.